Let's start recording. All right, so hopefully you guys got a little better score for the midterm exam 3. And whenever you have questions or any concerns or anything, just email me or come to the office hour or so. And there were a couple of concerns actually that students have addressed, asked. So I'm going to address that individually to those students. If you guys, some of you have emailed me and haven't, got my answers yet, I will. All right, so far we talked about all the macro, molecules, their structures, functional groups, how they interact, and the different kinds of macromolecules like lipids, carbohydrates, and things like that. So today we're going to actually use all those knowledges together, especially their structures. We're going to understand how the biochemical reactions actually occur and how they are regulated in the living system, especially using the enzymatic reactions. So these are basically for enzymes for today's lecture as well as the coming one too. So the enzymes that we're going to talk about are basically biological catalysts. And I believe I don't need to explain what the catalysts are, but they are basically one that catalyzes or make it faster or more efficient of the interconversions between macromolecules and their properties. And these macromolecules include, of course, proteins, as well as DNA, RNA, lipids, carbohydrates, and things like that. Pretty much everything in the living organism. And the enzymology is basically studying the biochemical reactions to understand how they are catalyzed by enzymes and their mechanisms, how enzymes work for the boosting those reactions, as well as changing the rates and the regulations for them, and so on. And there are basically two big sisters, or the two mainstream of the studies for enzymology, which studies the mechanism of the enzymes, which are structural biology, that studies the structure of macromolecules and interactions. And we've actually touched on a little bit of that, right? Looking at the structures and how that is related with their functions and so on. And the other big stream is the kinetics study, which studies the rate, the speed of the reactions and how they are regulated. And both of these, structural biology and kinetics, are important and contribute hugely to understand the reaction mechanisms that are boosted by the enzymes or the catalysts. And these mechanisms basically address reaction steps, participants, like reactants basically, group movements like functional groups, how they are regulated, how they're attached or detached from the substrates and so on, and their chemistry in enzymes active site, where all the biochemical reaction occurs. And combined use of these two ones, structural biology and kinetics, basically is fundamental for drug design too. It not just simply applied for the any kinds of substances that are useful for like cancer treatment and things like that, but also for the vaccines and many other things, basically, as long as they're dealing with the macromolecules in the living system, then you should study kinetics, structural biology, to understand how they are regulated in terms of their reactions. So before we get to all the enzymology or the enzyme functions and regulations, we want to basically cover the basis of the catalysis, starting with the catalysts here, which are the substances or the compounds or whatever that is that induces the biochemical reaction. So that as long as something speeds up the rate of the reaction, it can be called as catalyst, but it should not be consumed or used or altered during the reaction for as a reactant or the product. It's just that should be involved in the boosting the reaction speed. That's all. And catalysts, all the catalysts, including biological enzymes, do not change KEQ, which is equilibrium constant. And we already talked about this, right? What KEQ is, and we have the formula here where KEQ is related to delta G naught. So that's where we talked about all the thermodynamics basically. But we can make a direct relationship between KEQ and delta G naught. And delta G naught is again a Gibbs free energy for any kinds of reactions in interest at the standard state. So again, catalysts, including enzymes, do not change KEQ or delta G naught. So what does that mean? Will that change what? Or does not change what? I mean, KEQ, what does it define? And delta G naught, what does it tell you? At the equilibrium, that is correct. So that's what the KEQ is about, right? At the equilibrium, what would be the ratio of the reactants versus the products? But what about delta G naught? Basically, the ratio of that tells you one thing, and what is it? Can you speak up? Exactly. So spontaneity of the reaction, right? So the catalysts will not affect the spontaneity of the reaction. Even if you have a lot of catalysts, that doesn't mean that now you can suddenly drive that reaction forward or the reverse. It will be basically determined by the delta G naught, which is a sort of internal values or the states that will not change at all. But it just influences how quickly the speed of the equilibrium, those reaction reaches the equilibrium. And again, I talked about the relationship here. And enzymes, again, can only affect the kinetics or the speed, but not the thermodynamic properties like delta G naught or the KEQ. And an example of that, the catalyst working is the glucose, where you can see the reaction here. And is it endothermic or exothermic? Exothermic, because you have a positive values as a product here, so you actually basically emits the energy out in the form of heat, and that means also you have a delta G naught as a negative value. And what does that mean? Does that mean what is it? Spontaneous, meaning that exergonic, right? We talked about exergonic, endergonic reactions. So this is an exergonic reaction, highly exergonic because it has a huge negative value for its delta G naught. But it does not occur outside of the living system, actually, in most cases. And that is because even if this is a spontaneous reaction, it's not about the speed of the reaction. It's only the spontaneity, right? And the enzymes that are involved for this biochemical assays or the shift from the glucose to this end product happens much more in a faster rate inside the living system because of the catalysts, the enzymes. Again, it does not change these values, delta G naught or the heat that is excreted, but it only changes the rate of the reaction. So please don't get confused in between the rate of the reaction and the spontaneity of the reaction. Okay, so basically you can tell whether this reaction is spontaneous or not, or how it actually changes during the course of the reaction in terms of its energy state by looking at this reaction diagram. And it actually shows the uncatalyzed reaction, so the reaction without catalysts or the enzymes, and starts with the A and B as reactants, and basically gets to P plus Q as products here. And here are the G value basically, and it shows the delta G for the reaction. So is it a spontaneous reaction or non-spontaneous? Anyone? Is it a spontaneous reaction? How come? You don't know? that's most likely. Well, because of this huge curve here, basically, there's a huge hill here. Overall, this is a spontaneous reaction because when you talk about whether this is a spontaneous or not, you only talk about the final state. right? The initial state and the final state. It doesn't really care. Let's go back to the thermodynamics. We talked about all different kinds of energy state functions, but we don't care about the individual steps, the intermediate steps. We only care about the initial state and the final state, right? When you make a comparison between A plus P and the P plus Q, it goes down. So you have a negative value for the delta G. Is it correct? Because it's going down, right? So it is a spontaneous reaction. But like you actually pointed out, it has a huge hill in terms of this reaction, which makes this reaction not as much like spontaneous as like without any kinds of hill here. And this hill of the energy change is called the transition state. And that is basically, you have a lot of molecules for A and B there. They're colliding and making reactions and things like that. But individual molecules have a different state of energies, like different amount of internal energies. Some of them have a lot, some of them have just less of it. But some of them happen to have enough energy to get to the transition state, which is up here, will actually flow through this reaction diagram to become the P plus Q, as opposed to something which is right here in the middle, will not have enough energy to go through this, the curve. So it will be mostly likely goes back, and hopefully, by collision or something like that, or adding the having some energies or hit can recover, not recovering, but the overcome this transition state, and then goes to the final product. So this transition state is depicted as X double dagger in many cases, as you can see here too. So there's energy differences between the initial state A plus B and the top of this transition state X double dagger, which is called delta G double dagger. And that is basically a free energy of activation, which is different from free energy of reaction, right? This free energy of activation, delta G double dagger, is only for the molecules to reach the top energy state, the highest energy state, to go to the P plus Q, the product state. Any questions so far? Okay. So let's just consider a simple reaction here where there's a carbohydrate, the simple sugar here, and there's a conformation or change from A to B, from the boat conformation to chair conformation. And we already talked about this, where the chair conformation is a little bit more favorable in terms of its energetic state, right? Because it has less clashes. Okay, I expected this because the battery was dying, so it actually literally says dead battery. So that means I'm stuck here. So basically, there's a change between boat conformation here to chair conformation, and you can just take it as a biochemical reaction, where the initial state will be here as a G1, and it goes basically up to the this sort of intermediate state, which is a transition state, and then eventually it goes down to the chair conformation, which has a lower energy. So the delta G is again will be a negative value here. And again, transition point will be the highest energy point here, as we've seen for the sort of generic reaction curve here. And this transition state will be corresponding to a half chair conformation, which is the highest sort of energy because it is not the most stable form. It still has some steric clashes with their side chains, or the functional groups that are attached to the other carbons nearby. So it'll actually basically push away, but you have to have this state to get to the chair conformation. So it just have to happen there, and again, it's all about all the molecules or this both formation coming up to this energy state to become the more stable form. And this is actually important where you, in most often cases, have this transition state because it ensures the reaction does not occur always. So if you don't have this kind of, heel of the energy, but everything's flat, then naturally everything of the reactants will go to the products, right? Without any stoppage, basically. So you're gonna end up having everything here, but it'll be really hard to regulate the reaction rate or some kind of mechanisms involved there to going back and forth for the equilibrium, the regulations on equilibrium of those reactions and things like that. So it's always more convenient to have this to because it's all about the giving more energy there to proceed this reaction. All right, so we just talked about the how energy changes during the reaction. And again, the catalysts and the enzymes are all about the rate of the reaction, right? Whether it goes faster or not. And the rate of reaction is written as V for velocity. And this rate of reaction is proportional to the fraction of the molecules having enough energy again to reach to the transition state. So if you have more molecules having enough energy for reaching the transition state, then you're going to naturally have more reaction occurs at the same amount of time, right? So you're going to have a faster rate for that particular reaction. And this fraction of the molecules having enough energy is described by Maxwell-Boltzmann distribution, which shows this kind of Poisson distribution, looking similar to the normal distribution, but sort of right skewed here. And it basically indicates the number of molecules or the fraction of the molecules against their kinetic energy there. So when you look at this curve, and you can take it as a sort of a histogram, where it shows all the energy, the molecules have the energy points there. And this particular curve shows that the quite a lot of fraction of the molecules having some sort of let's say intermediate amount of the kinetic energy. So this is not enough to reach the transition state, as opposed to some of them, even if small fraction, but have the high kinetic energy here. And depending on where your transition state is, you can tell that how big, how much of the fraction is ready, or the going through the transition state to get to the products. So if you have a higher activation energy, and free energy of activation is same as again delta G double dagger here, and higher activation energy, meaning that you have a steeper curve, or the bigger curve, the heel here, and that means you're going to have even smaller fraction of molecules happen to have enough energy to go reach in this transition state. But if this is a lower energy activation energy, the reaction has the lower activation energy, then then basically this curve is much flattened out, right? So you're going to have more fraction of the molecules reach the transition state. Question? It's kind of hard to see you guys with kind of this microphone, but any questions? Okay. Is it all clear, or like? And when you look at this one, it actually shows the same curve here, Maxwell-Boltzmann distribution. But you can also change the temperature here, which will input the additional energy to the molecules, right? So with the higher temperature, which is black curve here, then naturally more molecules will have higher kinetic energy. Like you're going to have more Brownian motions and more collisions and so on. And naturally, you're going to have more fraction of the molecules will get to the transition state. Hold on. I actually skipped the quite a bit of explanation here. So this rate of a reaction can be described by Arrhenius equation, which shows the K, and K is rate constant of the reaction. It's not the rate of reaction, it's rate constant, which is proportional to the coefficient in the rate law. So that means if you have a higher K value, that means you're gonna have higher rate of reaction. And this K is defined as A, which is constant specific for a given reaction, times E, and it's an exponential function for the E, with a negative delta G double dagger divided by RT. And here R is a gas constant, as you know, so it's a fixed value. Temperature can change, as well as delta G naught double dagger is defined by the reaction that you're looking at. And A is also dependent on the specific reaction. So some reactions will go much faster, right? So in which case you're going to have higher A value. And this Arrhenius equation, you can basically estimate the rate of the reaction V, which is a product of K and the concentration of the reactants. And this S is for the reaction from S to P. You can take it as the carbohydrate conformation change, or some kind of reaction there, whatever that is. I mean, they're going to be always the same equation applied here. And when you take into consideration of this Arrhenius equation, the bigger delta G double dagger here, which is in exponentials, but with the negative value. So that means the bigger the delta G double dagger, you're going to have the smaller the K value. And when you have a smaller K value, that means you have lower V too, smaller V, and that means a slower reaction. Does that make sense to everybody? And basically, you can increase this K to speed up your reaction too. And to speed up the reaction, again, you can use more substrates or the more reactants, as well as you can increase the K. And there are two ways to increase the K when you look at this Arrhenius equation. The first one is to decrease delta G double dagger, right? Again, this is an exponential with the minus here. So the smaller the delta G double dagger, the higher the K value. And also you can raise the temperature here. If you raise the temperature, again, you're making the smaller value for this negative exponential. So you're gonna get higher K value, right? And this number one and number two, lowering the delta G double dagger or the raising the temperature, increase the fraction of molecules possessing the sufficient energy to get to the transition state. So it's all about how many fractions of molecules to reach transition state to make the reaction faster. And for many chemical reactions, rate increases twice much by increasing temperature by 10 degrees C. And you can simply just plug those numbers into here, right? And also the catalysts, as well as enzymes, decrease in this delta G double dagger instead of changing temperature to have the higher value for this K to make the reaction faster. And you can imagine that temperature doesn't change too much, especially in the living system, but you definitely can lower the delta G double dagger. And that is about the lowering these differences between the catalyzed reaction and uncatalyzed reaction, especially for this delta G double dagger value. So the differences of delta G double dagger value between uncatalyzed one and catalyzed one will be written as delta G double dagger. Make sense? It's going to be changes of delta G double dagger. And you can put cat for the catalyzed reaction. All right, so we're keep talking about the rate of reaction and using this Arrhenius equation to understand better how catalyzed reaction boosts the reaction speed. And the degree of rate enhancement can be calculated by looking at the different rate constant values for the uncatalyzed and catalyzed reactions. And that is simply the ratio of catalyzed one divided by uncatalyzed one. So naturally, it means that if you're going to have a much faster speed for the catalyzed reaction, then you're going to have a much higher K2 value, right? So this ratio will increase. So your degree of rate enhancement increases too. So let's start with the simple Arrhenius equation here. K equals AE, negative delta G double dagger divided by RT. And again, reaction rate is V equals K and your reactant concentration. And for uncatalyzed ones, we can simply just put 1 for the K as well as delta G1 double dagger instead of just the delta G double dagger. And same thing applies for the catalyzed reaction. We put number 2 for those to distinguish them. And again, degree of rate enhancement is the ratio of those K2 and K1, right? So we basically use the same equation here on the different, the other side and put them together here to make the relationship. And because shares the same A cancels out, and use the same base for the exponential. So you can actually put it together because this is division. You can put minus between the exponentials to get this. And delta G1 double dagger is right here, which is a transition state, the free energy of activation for uncatalyzed reaction. And delta G2 double dagger would be right here, which is I actually incorrectly put 2 deltas here. It should be just the 1 delta and 1 delta here too, as you may have noticed. It's gonna be the free energy of activation for catalyzed reaction, and basically you subtract this delta G2 double dagger from delta G1 double dagger to get the delta G double dagger. So you replace it here with this value to get the final relationship between the degree of rate enhancement and the delta G double dagger. Okay. And this delta G double dagger is basically difference between these two, the delta G double dagger values between uncatalyzed and catalyzed reaction. And you can also put cat here for catalyzed reaction. And since this is an exponential function, small changes in delta G double dagger will lead to a huge changes in degree of rate enhancement. So these are 2 examples where only the increase of a 5.7 kilojoule per mole, which is much smaller than 1 hydrogen bonding, can give you tenfold of rate enhancement. And if you plug in the 34 in here, which is about 2 hydrogen bonds, then you can achieve 10 to the 6th rate enhancement. So with the small changes in delta G double dagger, you're going to have a huge changes in the rate enhancement. Okay, so far we talked about the catalysts and the catalyzed reactions and get the rate of speed or rate of the reaction for the catalyzed and uncatalyzed ones. Now we're going to move on to the enzymes, which are the biological catalysts. So they also accelerate the rate reactions through lowering the delta G double dagger. So it's the same as just the regular catalysts. And also work via combining transiently with the reactants. So if that is a permanent work, more like the long-term kind of reaction, then it'll be considered as a reactants, right? But they are not part of reactants. They are just enzymes or the catalysts. So they should be just have the interaction transiently with the reactants as well as products. And they promote their entry into the transition states, which it requires the high energy, and that's the biggest hurdle for the reaction. And enzymes are known to be the best catalyst on Earth, and there are several differences between chemical catalysts and the enzymes. The majority of biological enzymes are proteins, although some of them are RNAs or even RNPs, which are ribonuclear proteins. But chemical catalysts are simple acids, bases, or metal ions in many cases. And typically, enzymes are more efficient. So when you just compare the reaction rates, the enzymes have a much higher reaction rate, 10 to the 6th to the 10 to the 12th times higher than the uncatalyzed ones. And also, when you make a comparison with the chemical catalyst, it has a huge difference here. Can be 100,000 times to the much more like efficient, achieving the higher rate of the reaction compared to the just the regular chemical catalysts. And also the enzymes work under the mild conditions like a 37 degrees C in the most of like living systems, and under 1 atmosphere and the neutral pH like around 7 or so, as opposed to chemical catalysis happens mostly at the extreme conditions. And also enzymes have a greater specificity to substrates as well as reaction types. Examples is that the many of the amino acids, sugars, and so on, all the many macromolecules as well as some simple molecules that are used for in the living system have a stereospecificity. So they can have either L forms or D forms, but mostly have certain form, which is L form for the amino acids, right? And the reason is because all the enzymes are involved in a way that it chooses only one form there. So it has a stereospecificity for there, which determines the abundance of their products as well as substrates too. And enzymes typically have just one substrate for one enzyme, which is quite specific and quite focused. And also there are no non-productive side reactions. So it only have its own particular job, and that's pretty much it. Does not have any side products in most cases. And enzymes can also be finely regulated as opposed to these chemical catalyst acids, bases, and metals are just have to be there, or you have to just add them there. And that's pretty much it, right? It's hard to get rid of them unless you induce any other kinds of reactions. But for the enzymes, since they are just the proteins, they can be altered in their expression level at the transcription level or the translation level, going through the protein synthesis and degradation. And you can basically change those to regulate the amount of enzymes that are available in the cell or the living system. And also you can change the enzymatic activities too of the existing molecules or the proteins by putting some covalent modifications like phosphorylation or the smolylation and so on, as well as you can use various classes of inhibitors and activators too that are also in many cases just proteins. And also that includes the allosteric inhibitors and activators. And allosteric inhibitors or activators are basically not working directly on the active site, but working on the different site rather than active site to change or induce the changes in the function or the efficiency of active site. So sort of working indirectly, but that is used quite often in the living system. And enzymes, again, is basically speed up the reaction by changing the transition state, right? So it reduces delta G double dagger, which is, again, the free energy of activation through binding to the substrate to form the ES complex. And this binding occurs at the active site. And active site is basically sites within the protein, within the enzyme, where the actual enzymatic chemistry takes place. And This binding takes place through the non-covalent forces in many cases. Again, if it is a covalent or permanent binding or the changes, then would have been just the reactants, not the catalysts or the enzymes, right? And many of these forces are the same type of forces that hold together the protein structure too, like ionic or electrostatic interactions, hydrogen bondings, van der Waals interactions, like hydrophobic interactions or dipole-dipole interactions, and so on. And enzymes usually have a distinct cleft in the active site to have a specificity um, where the substrate, specific substrate fits in there. And it can use many different kinds of properties there for those interactions, but mainly used in two big aspects of complementarity between enzymes and substrates: geometric complementarity and the electronic or the electrostatic complementarity. As its name says, geometric complementarity uses the contour, right, the shape, interfaces that those two molecules interact to each other. So that's gonna be convex or concave, those kind of specific shapes, and the size of the blobs that is protruded or um, uh, uh, um, recognized by the enzyme. So basically works as a knobs into holes, and electronic or electrostatic interactions would be basically using the charges. So positive charge to the negative charge and so on. And it also includes the hydrophobic interactions too. And let's say that, so it is great. I mean, probably through the evolution, it try to find the better and better and more efficient binding between the enzyme and substrates, right? But does tight binding to substrate mean that the enzyme is the most efficient? Is the tight binding a good thing or bad thing for an enzyme? You can guess. I mean, it could be a good thing or bad thing, but would it be a good thing? Yes. Right. For stopping, I mean, so um, it can be both basically, but for stopping the reaction, you can regulate the enzymes at the translation or transcription level, right? We talked about some post-translational modifications or the degradation of a proteins or synthesis of a proteins that can actually have, work as a mechanism for regulating the enzymatic activity there. So tight binding, can be a good thing, I guess. Is that a good thing? No? Mm-hmm. Any other thoughts? Interesting. So that is actually absolutely true. So the last point that was made was about the reversible reaction. Whether what do you think? I mean, if enzymes there, do you think the so it should accelerate the forward reaction, right? Reaction to the forward direction. What about the reverse direction? Would that enhance the that too, like that reaction too, the reversed reaction too? No, why not? What does enzyme do? It lowers the activation energy. So basically lowers the little bar that the one thing, the reactants goes to product. What about the reverse reaction? The product has to go to the um, reactants for the original reaction, right? But when you imagine that, that also you can consider that as a huge kind of, I don't see the pen here or marker, but it will have a much larger activation energy or the transition state it is required, but it's lowered down. So basically, adding the enzyme won't change the delta G or KEQ, meaning that you're going to have the same number of and what is it the products concentration as well as reactants concentration. It's all about the speed, right? So you're going to actually boost the reverse reaction too, both forward and reverse reactions. So you won't only change the what is it the KEQ, equilibrium constant, but speed is accelerated. Going back to the original question, is the tight binding a good thing for enzyme or not? It's a bad thing. It is a bad thing because, I mean, you want to have a tight binding for making the ES complex, which is a enzyme and the substrate binding at the transition state. But once it is reached that now you want to just let go of your substrate, right? Because it's not a substrate anymore; it's a product. But if you have a tight binding, you're going to still hold on to that. So the reaction cannot be occurred with for the other molecules. So you have to have a nice balance where you have a pretty good specific binding there, but still it should happen only during the transition state, during the ES complex formation. But after that, once S the substrate changes to product, you want to let go much faster. There are two models to explain how enzyme works for binding their substrates as well as the catalyzing the reaction. Those two models are the lock and key hypothesis and induced fit hypothesis. The lock and key hypothesis, which was proposed by M.O. Fisher long time ago, explains the substrate specificity where the substrate have the specific shape and the enzyme recognize that specific shape because it has that matching kind of curve there, matching kind of interface there. Again, it explains pretty well about the specificity, but it does not say anything about why the catalysis actually occurs. Because it's only the binding, right? Substrate binds there and there's nothing more happens there. As opposed to induced fit hypothesis, at least partially, explains the how the enzymatic reaction occurs here. So where the enzymes still have a binding site or active site there, but it was a little bit distorted. And to make the tight binding between the substrate and enzyme, the enzyme has to be distorted too a little bit. So enzyme can the substrate. So enzyme can induce the distortion of substrate a little bit to make this ES complex basically. And then once it's changed a little bit or distorted, I'm talking about the substrate, then it can become its product or making the reactions much in a faster or efficient way. This is a really kind of well explains how the enzyme works for this catalysis. But the problem is that in reality, enzyme is not distorted, actually, in most cases. But they have been evolved to bind the substrate in the different ways that we're going to talk about a little later. But sometimes actually enzymes undergoes conformational changes, distortion, if you will. And an example would be this enzyme, which changes the glucose to glucose 6-phosphate. So this enzyme is called hexokinase because it binds to the glucose, like basically, any kinds of like with the hexo ring, And it happens during the glycolysis. It's the part of the metabolism. And that happens basically by this kind of fitting between the enzyme, the shape of the enzyme and the shape of the glucose here. So you can use the lock and key hypothesis for this binding. But it's unusual because it experiences a huge conformational change on this enzyme. So you can take it as a sort of induced fit, hypothesis where it changes the shape or to make the distortion of the enzyme, as well as induce a little bit of this substrate too, make this, make it into the glucose 6-phosphate. But again, it only happens sometimes. Not only so not all the enzymes go through this conformational change. Also, there are many other proteins, especially structural and motor proteins, to use in this kind of strategy where they go through the conformational change for their cycles to make the higher order structure of the protein complex or the binding to a certain kind of different substance. Okay, so we'll keep talking about the enzyme specificity, and it can be largely due to the lock and key principle that we already talked about, have the matching shape or the matching charge, and so on. And again, it affects the stereospecificity, where it has a certain isoforms, stereoisoforms that can interact with or the making as an output too. And there are different degrees of geometric specificities in different enzymes, and that determines the efficiency of those enzymes or the specificity of the substrates. And often enzymes will just not only act the one specific substance, but will act on the functional group of a similar structure. So the many enzymes can be pretty much like promiscuous in their substrate. An example is chymotrypsin that cleaves the peptide bond with the any bulky hydrophobic residues. So as you can remember, bulky hydrophobic residues like tyrosine or phenylalanine and so on, you can actually detect all those kind of things and it will cleave the ester bond for the digestion. Enzymes have different names, and that that nomenclature was coming from the sort of trivial names that does not actually have much meanings, but can be used in convention, basically like a catalase, or it can have a more systematic names too, where it uses a substrate name and put the ASE, basically A's after that, with the classification number, which comprises 4 numbers and connected by dots, like 1.1.1.1, something like that. But typically, the enzymes are named by the substrate and the A's without indicating classification number too much. An example is lactase here, which breaks down basically lactose. And as you may know already, lactose is a disaccharide of glucose and galactose. And this IUBMB, which gives the classification number, classifies enzymes also based on their reactions, particular biochemical reactions. So here are the examples. Oxidoreductases are for the catalyzing the oxidation reduction reactions or redox reaction. And transferases are basically transferring or moving some kind of functional groups there. Hydrolases are inducing the hydrolysis reactions where water molecule is involved there to breaking some like their substrates. And lyases are basically eliminating some double bonds there. Isomerases for isomerization, ligases for fusing together some functional groups or molecules, and so on. Question? No, it is still widely accepted. Lock and key is still valid. And it really explains well about the specificity of the substrates. it only does not explain how the enzyme, enzymology, enzymatic actions actually work. So that's it. But it is a still great model that works for pretty much everything. Mhm. Right. So it's about the specificity where you can have multiple, targets or substrates that you can use. But whenever enzyme works, it works with just one enzyme at a time. So that's the how reaction occurs. So regardless what kind of substrates you're gonna have for that enzyme, there'll be only one in most cases, as opposed to that one substrate can be many different functional groups sharing similar properties or the structure. All right. So now we're gonna get to the some examples of these enzymes, especially serine proteases or the serine endopeptidases, and they are really important because they're really highly abundant in the living system, as well as they're involved in many different kinds of biological processes. So these are serine proteases. As its name says, it has the serine in the active site of the enzyme, and they can be grouped into clans to subgrouped into clans. They share the similar structural properties or the structures, and also those clans can be subgrouped into families that share the close sequence homology. And the major clans, and we're going back to this, the clans that shares the similar structures. Um, the major clans are chymotrypsin-like, subtilisin-like, the alpha-beta hydrolase, and signal peptidase clans. And we're going to mostly focus on this chymotrypsin-like clans for this lecture, which are really important because they are having a critical role in metabolism, especially for digesting the proteins. And those all serine proteases participate in a wide range of functions in the body, like blood clotting, immunity, inflammation, growth, and so on, signaling too. And also, of course, contribute to the digestive enzymes during metabolism. I talked about chymotrypsin-like for chopping down the proteins. So let's delve into this chymotrypsin-like serine proteases. They have three major proteases: chymotrypsin, trypsin, and elastase. And again, we're going to talk all about them, like their properties and their substrates, the specificity, and so on. These all three are synthesized in the similar place, the pancreatic acinar cells, and secreted into the small intestine. So basically, that's for digestion, right? And all three are similar in their structures that was validated by the X-ray crystallography. But the differences is in their substrates. Specifically where on the polypeptide they're cleaving, make the cleavage. And those like peptide bonds that are cleaved are called cis bonds. So that means these three different serine proteases have different cis bonds for their targets. And each of these proteases target different regions of a polypeptide chain, especially looking, and that property is determined by the active site of the serine proteases, how those, amino acids, what kind of amino acids are forming the active sites, as well as how those side chains are exposed to the active sites or arranged there. So here are the specificity of those three chymotrypsin-like serine proteases. Chymotrypsin, which shows its active site in this cartoon here, is responsible for cleaving the peptide bonds with the bulky hydrophobic amino acids. Like I talked about tyrosine already, but tryptophan, phenylalanine, methionine, and so on. Anything bulky, but also hydrophobic. Because the this hydrophobic side chains can be snuck into this hydrophobic pocket of the active site pretty nicely. And here's another different diagram here for chymotrypsin here. Um, it has the serine, obviously here, which is critical for forming this active site and recognition of their substrate. But basically phenylalanine or any kinds of things that have a bulky region, hydrophobic region, because of these glycines, can be easily snuggled into this cleft. And there's going to be a cleavage right after phenylalanine. So again, this is going to be called as cis bond. Trypsin prefers lysines and arginines, and here's the shape of the of the active site of trypsin. And what are the common features of lysines and arginines? What is it? Lysine K, arginine R. Do they have common feature? Exactly. So they're basic, right? Basic amino acids, or the amino acids with the basic side chains. So they have a positive charge. That's why there's an aspartic acid sitting right on the middle and the bottom of the active site in the trypsin. And aspartic acid, because it is, as its name says, it's acidic. So it'll attract the basic side chains, right? So it's, and of course, acidic is negatively charged, basic is positively charged. So it's going to be the geometric matching as well as electric matching too. Similar thing can be applied to elastase here, which prefers alanine, glycine, valine. And what do you think about the common properties of these amino acids? Nonpolar, but also small. They're small. Alanine is just a methyl group, glycine is just hydrogen, valines like isopropyl, right? So it's really small. So elastase have a really shallow active site. And you can see that because of this valine and threonine that is blocking the active site here, so only the small amino acids can be sitting there. Small amino acid, I'm talking about the small side chains. Here's a catalytic mechanisms of the serine proteases. And the main player is called catalytic triad in the active site, and it's located in the active site and where the catalysis occurs. And also this catalytic triad is conserved in all serine proteases. So you're gonna see the same sequence in all serine proteases. And a coordinated structure consisting of these three amino acids, histidine, serine, and aspartic acid at this specific position or the residues. And they're located very close to one another near the active site. I'm just reading this here. And play a critical role, essential role for cleaving the cleaving ability of those serine proteases. And how it occurs is basically kind of ping pong catalysis. So first, the substrate binds to the enzyme here, and substrate is the polypeptide basically, right? For serine proteases. And the product, the half of the product will be released because of the cleavage on the cis bond. And then they're gonna be basically ES, still the enzyme will be bound to the other half of the polypeptide. And then another substrate, which is a water molecule that comes in here to that complex to make the another half of the polypeptide leaves. And the enzyme will be recovered as its original form. So now it is also available for the other substrate, which is a polypeptide, for another cleavage. And each amino acids within this catalytic triad performs the specific task. So they are really required and have to be on that specific position for the most efficient cleavage. And this process is called the charge relay system, where it basically relays the charge, one charge there. First, when you look at the serine here, 195 here, it has the hydroxyl group here. And because of the hydroxyl group, it will actually release the proton or the hydrogen cation here, right? And that can be boosted by this histidine 57, which is basic, right? Since this is a basic, it prone to accept the proton, right? So it'll pull the hydrogen from serine, make this serine more negative, right? And this negative charge can easily attack this carbonyl group of the polypeptide. And also this histidine reaction, which its job is to pull the proton from the serine, can be stabilized by this aspartic acid in the 102 residue place, because aspartic acid is again the acidic, so it's negatively charged. So it'll stable this basic side chain of histidine. So if you're missing one of them, then you're not gonna have enough electron relay system there to get rid of this high proton to attack the cis bond. Okay, so far we talked about the mechanism, how the serine proteases actually work. Now we're going to move on to the other state of the enzymes. There are zymogens in the living system, and they are basically inactive form, the precursors of the enzymes. And they're typically much larger than the actual active enzymes. And they can break into parts or reveal some kind of important parts like active sites or so after cleavage. And they are basically taking the strategy where packs with the inactive bigger portion of the enzymes, because if the enzyme itself is expressed or the translated right away and have the activity right away, it'll start chew up the things like around when they actually meet the substrates, right? But many times the protein synthesis, including enzyme synthesis, happens in the ER, and you don't want that cleavage or the digestion happens in the ER, right? You have to bring it to the right place, like intestine or so. So you just have a bigger package, like unpacked ones, especially hiding the active site in the form of zymogens first for your expression, and just deliver it to the where it has to go. And then once it gets there, now you can reveal the active site by like having the confirmation or change of the zymogens, or especially make the cleavage of the zymogens to expose the active sites. So after that, there's a confirmation structural changes of the zymogens, which will basically make the active site available or open for the proteolysis. And certain enzymes require cofactors for their full functionality. And because when you think about the enzymes, they're proteins, right? So they can have a lot of different conformations for their constituting amino acids on the active sites, but still it is limited because everything has to be start from the side chain properties. So it can be either like polar, non-polar, charged and things like that. It's pretty much limited to the side chains. So to give the more flexibility and the more diversity in terms of their reactants or the substrates, they have to recruit the other things like non-organic stuff. So they are called cofactors here, and that broadens the range of enzymatic reactions or the properties, and basically serves as a chemical teeth, right, with the different sizes and different shapes. There are different types of cofactors. The metal ions are widely used, and also coenzymes. They can be simply prosthetic groups, like simple groups, or can be small proteins, small polypeptide too. And they basically change the chemically during the reaction and must be regenerated on a different protein. So because these coenzymes are not the product of any reactions that it is involving, or it's not the side product of the enzyme itself. It has to be generated somewhere else and can be bound to enzymes and then will be regenerated in the other process to keep providing that for the full functionality of the enzymes. And especially the prosthetic groups, the small functional groups, are tightly bound and often covalently bound to the organic groups to make the enzyme active. And enzymes, especially which involves the cofactors, are called holoenzyme, which is an active form, and that is basically a Apple enzyme, which is an inactive form without cofactor, and the cofactor. So when you put it together, making into active form, again, that'll be called holoenzyme. Any questions so far? Okay, so these coenzymes can be classified by their sources. Largely, there are two classes. Metabolite coenzymes, as it says, these are metabolites. So you can actually generate that during the metabolism in the living system. And the other class is vitamin-derived coenzymes. So those are vitamins, and in many cases, has to be uptaken, from the outside, from the other source. So metabolite coenzymes are synthesized from the common metabolites like the sugars or the proteins and so on. There are two examples here. One is like nucleoside triphosphates like ATP or GTP and so on. And they're widely used because they contain a lot of energies there, right? So they can be used as an energy source and can donate the phosphoryl groups in multiple times there as a sort of a prosthetic group too. So for the phosphorylation or some other reactions where the energy input is required. And the other one is uridine diphosphate glucose or UDP glucose, and also the S-adenosylmethionine. And this UDP glucose is a source of glucose for synthesis of glycogen in animals and starch in plants. So it's just an example, how this is used as a coenzyme here. And another one, S-adenosylmethionine, can donate the methyl group because it's based on methionine, and can be used in the biosynthetic reactions. As opposed to metabolite coenzymes, which you can actually get within the system, there are vitamin-derived coenzymes. Again, that's typically, you have to just intake or uptake from the other food source. And they're required for the coenzyme synthesis too, so they can work as a precursor for the coenzymes. And of course, lack of certain vitamins cause certain diseases. And there are two subcategories in vitamins, especially used for making coenzymes: water-soluble and water-insoluble or lipid-soluble. And these are the classes, basically. Vitamin B and C are the water-soluble ones, required in the daily diet, but the excess one can be excreted in urine because this is water soluble, right? So basically you can just dissolve it in the urine. As opposed to lipid soluble vitamins have to be limited because if you take a lot more, then it cannot be easily excreted. It will be deposited in the fat for a long time and cause some problems. Again, many vitamins are coenzyme precursors, so must be obtained in from the diet. And these are the examples that you're probably familiar with quite a few of them here, like a biotin, riboflavin, folic acid, or ascorbic acid, and so on, as well as these vitamin A, D, E, K, and so on. And they have the matching coenzyme products here, which is important for enzymatic reactions. And these are what kind of reactions that are involved for that, as well as there's a relationship for human deficiency disease like scurvy or hemorrhage and so on. Especially cobalamin, which is right here, B12, is enriched in organ meat like a kidney and liver. Riboflavin is enriched in milk, in whole grains, livers. Nicotinamide is important, but it's rich in cereals, meat, and legumes, and the folic acid in green leafy vegetables, as well as liver and yeast too, and thiamine too, in basically many kinds of different kinds of food source, right? And the message here is that you need to eat well, with a lot of diversity to keep you well. And that's it. So